Morning, everyone. Thanks, it's good to be here. What I'm going to do today is I'll just go across some of the research that we've done over the last while. A lot of this is done with Dion, who's here in the audience. So despite decades of research on COTS, there's a lot of key traits that remain to be characterized. And if we're looking through the lens of echinoderm evolution, and particularly C-star phenotypic plasticity, points to areas that might be fruitful to investigate, traits to look for that would maybe fill those pieces of the puzzle to understand how successful and why COTS is so successful. The paradigm, and we all know that in order for COTS outbreak to happen, you have to have successful larvae and successful juveniles. And this is, most of the research we've done have been on the early life stages and a little bit on the reproduction, but I'll just mention this in passing, as I will with the others. A lot of this is already published, but just to point out a couple of really bizarre things that we found with COTS over the last while. So this is a spawning in one tree lagoon. This is a male that's surrounded by sperm. There's a female with eggs, and that's another female. So this is that happens in sort of the late afternoon. But then if you have a look at some of the eggs that the female releases, it's quite, we see occasionally what looks like a self-fertilized egg. Now, what does that mean? We're not really quite sure. But we have also found from looking at some of the transcriptome work, we couldn't have done this without having the genome available, that there's indications of cryptic hermaphroditism. Now, this is really unexpected, where you've got females expressing male genes and males expressing female genes. And so we were delighted to get all 8 gamete recognition proteins isolated for COTS. And this shows that this is phenotypic plasticity. Now, I've done lots of starfish, lots of sea urchins with this. This is unusual. This is not typical of spawning echinoderms. So you have the potential for microhermaphroditism, maybe reproductive assurance in isolation, and it could explain why Ross Babcock found in 84 paper that he found pretty much an isolated female COTS having high levels of fertilization. That could have been self-fertilization. So I think there's some flexibility here with what COTS might be doing in the real world. That creates a problem, but we probably need to know if it's possible. Now we'll move on from that to the larvae. So for the larvae, I'll talk about today 2 areas that we've concentrated on, and that's cloning and the microbiome. And how does COTS compare with other sea stars that do this as well? Well, we do know that the ideas of the proximate causes of outbreaks has focused on the larvae and the larval hypothesis. There's 2 aspects of this larval hypothesis. There's the larval resilience hypothesis, which I think has been largely ignored. This is Randy Olson's work, and there's something to that for sure, where he grew them in Visit Island Lagoon way back. But there's also the larval starvation hypothesis, the limited nutrient hypothesis, which is pretty much the paradigm that's been expected. And that was sort of linking to the terrestrial runoff hypothesis, that if you take care of terrestrial runoff, then you might actually help reduce COTS outbreaks. The link is actually quite tenuous. So then, having a look at larval cloning, we know that sea stars are really great regenerators. They split themselves in half, make more of themselves, etc. They do that both at the larval stage and the adult stage. And so the larvae are potentially, eternal. Sorry, I keep hitting the wrong one. So we know for a fact that we've done this in the Caribbean, so we know that the larvae are cloning. So in 2015, I decided I'm sure that COTS clones, and it does. And if you look at this paper by the Suzuki et al. In the 2017 issue that Morgan just showed, there's the bipinnaria that's budded off a little bit there, and here is a brachylaria with 2 clones on both sides of it that's just about to be. So the COTS are cloning in the plankton, for sure. And so if you want to have a look at how this compares to this Indo-Pacific species, just a recent paper, there's a little clone bud that's coming off this larva, and it's exactly the same. So the crown-of-thorns are cloning in a manner which is typical of sea star larvae. So they could be out there in the plankton for a long time making more themselves. So this is, and more about larval plasticity. They do it by two ways. The cloning, they can bud off a bit of themselves, and they can split in half to make a head and a tail. And this is so fast. It's very hard to catch, and they regenerate so quickly. Next time we're going to raise them at 24 degrees so we can actually slow the process down and watch it. But anyway, within a few days, you've got a complete looking developing larva, and 10 days completely. You would not know that this was a clone product without having followed the individual larva. So what factors promote cloning? Spent the summer raising clones, but what is interesting is that some mothers are just super cloners, so it's variable. Dad might be involved, I'm not sure yet, but so we've got, and the cloning varies between parents. And with food are as higher, they might make more of themselves. That supports the terrestrial runoff hypothesis. And they also will clone in the presence of a predator, so split themselves in half, maybe to make them smell smaller to a planktivorous fish, or just give the fish the half of them so they can regenerate the other half. And the other aspect is that they do clone in response to physical disturbance as well. So asexual propagation occurs. There's pictures of them from the plankton, and there's some indications that there's a signal of that in population genetics, and I'll get to that in a moment. And together with this potential, and I say potential, we have to figure this out, for reproduction by selfing really has implications for how you model sources and sinks of COTS populations, and this could have real implications for adaptive molecular evolution. These guys are so fecund, have so many gametes. You can imagine the evolutionary potential for such a highly fecund organism to have such variation is enormous. So to have a look at that then, this is some work we did where we simulated the COTS populations from the Great Barrier Reef. This is a work in review. Using the paper that was published in that diversity issue by Harrison et al. And if you actually model that population, I won't go through the details, but I can talk about that later, you can actually see the FST, the genetic signal, right smack in the middle, indicates about 12 pairs of clones. So there's an indication from population genetics from data from the Great Barrier Reef that cloning is, and potentially self-fertilization could be a signal in the reproduction of in the population genetic structure of COTS on the reef. So the next is the microbiome. Again, this is published, so I won't go into a great deal, but we do know that the COTS has a distinct microbiome, and similar to the cloning larvae from the Caribbean, there's some phototrophic bacteria residing in the gut cells, potentially providing food for the COTS. So that could be another indication of a tropical symbiosis. This is work done with Tyler. So so we've got, I'll just sum that up at the end. So there's a lot of interesting aspects of the larvae. Now we'll go on to some of Dion's work, which was on the juveniles. And as you know, there's two phases, the algal eating phase and the coral eating phase. So what Dion found was that they don't just eat CCA, and in fact they do quite well on various coralline algae, but what's really stunning is that they can persist if in poor conditions just on a biofilm for 300 days. And if you take so if there's nothing much in the way of CCA around, they can hang out. But if you then take that CCA, that juvenile, those juveniles that didn't do so well on biofilm, give them some ordinary, they do catch up, but it might take another year for them to get to the stage of the transition to coral. So this is our hidden army paper, that Dion's work. These are juveniles that we had for years in our system, so 6.5 years, and a 10-month one, which was the new. But if you actually look at the modeled growth of the two, it's quite similar. So the herbivorous juveniles can persist for at least 6.5 years before we transition those 6.5-year-old juveniles to coral. And when we did was they performed and they grew at exactly the same rate as a new group coming through. So you've got, so for instance, if these little that 6.5-year-old geriatric juveniles is what I call them. You can give them coral and they will just grow up fine. They will turn out from a Peter Pan to a reproductively mature adult, but they just took 6.5 years before they did the transition. So that's out there so the possibility is you have what we've called the hidden army. And Greg was showing some of these juveniles yesterday. So we know these juveniles are tough. They can survive on an algal diet, even a biofilm, for years. The juveniles, so therefore, if times are tough after a coral bleaching event and there isn't much food around, they could build up in the reef infrastructure, just waiting for good times. And then, and so this might explain that time lag between spawning and the outbreaks that we see on the reef. So the transition. So now, if we looked at the transition and these juveniles and how they fare going from coralline algae to a coral diet, and we monitored those juveniles for 3 months. And how did they go? Well, it's perilous for them. The coral fights back, and coral stings. So the poor juveniles, they really do have a tough time when they hit the coral. And so they have this avoidance escape for the juveniles to coral defenses. And they have a lot of damage in the first 5 weeks. About 30% of the juveniles are damaged. Some of them just don't make it. They die. They just don't survive. Those stings are fatal, whereas others, you can see the arms that are damaged. They do recover, but it takes months for them to recover. And so then to have a look at how they go in comparison. So here we have the undamaged juveniles and the damaged juveniles, quite a bit differences in sizes, and 50% of the juveniles that are injured did not complete their transition to coral for 3 months. Some actually went back to the algae to refuge and said, oh, I better eat algae again, that coral's too hard. And then some of those never went back to the coral again. They stayed as algal-eatal juveniles to the end of the experiment. But for the most part, the estimate recovery time after being stung is about 4 months. So the consequences of coral stings. Now, the interesting thing is that, just as an aside, Greg mentioned the bottle brush corals. They've got nice little grooves that the juveniles might hide in to protect from those sweeper tentacles. So it could be there's a reason, not just because they like that acropora, but because it's a place where they survive better. So very interesting observations yesterday. So we've got this, so the injuries may delay the transition to coral and kill the juveniles and certainly influence the population dynamics. So there's another aspect which is perilous for the poor juvenile COTS, is those coral guard crabs. And these juvenile corals can detect these crabs from a distance. And they have these incredible profiles that they make, a spine ball. And Motokawa showed this in a paper way back in 19, whatever it is, bottom here, that the ambulacral grooves, they zip them shut tight to hide those tube feet, because those coral crabs sit there and nip the tube feet and eat them. So, and this is another juvenile which folded itself in half. So the juveniles curl their arms or fold in half to protect the oral side from the crabs that pinch the tube feet. Morgan showed this. This is a big, this does happen with the adult COTS as well, but the juveniles are running the gamut of the guard crabs. So these guard crabs are really important as well. So then to have a look at just one little aspect of this, the writing response in the presence and absence of a guard crab. When they turn themselves into a protective ball, it takes them a little bit longer to actually uncurl than when the crab is present versus the crab absent. And this has potential for where the juveniles are in the reef as well. So just to add something from the adults, we've been having a little bit of fun playing in Montreux Lagoon with our COTS, and everyone have seen this. These guys, when COTS are disturbed, they curl their arms orally. Everyone's seen this, loads of pictures. Even when the COTS control guys pick them up, they curl in. And so what does that mean? They make these spine balls. They turn themselves into a sea urchin. And Dennis Devaney, Dennis, he worked a lot on echinoderms. A lot of brittle stars are named after him. That was his thing. He died diving off Hawaii in the 80s. But anyway, Dennis did this all through, with Randall, through the Indo-Pacific. And he identified this balling effect. And he showed that they were the balls would surge. So, Morgan, they might try and walk on their tube feet, but incidentally, they can pivot around the reef as a football or a spine ball. So then it might be a mechanism, not a mechanism intentionally, but it could actually displace the COTS around the reef, this spine ball. So lots of interesting observations with respect to the defensive mechanisms. When you look at them, there are just so many defensive mechanisms that it's really pointing to the fact that in the evolutionary history of COTS, predation pressure has been an important selective force in determining their morphological attributes that we see today. And as we were hearing yesterday, red emperor and various fishes really like to eat COTS. So it could be this, as Frederica might be here, but she was talking about some of her work as well, that there's way more to that predation hypothesis for the COTS than has been probably given credence. But it's emerging now, I think, from a weight of evidence from a lot of people's observations. So I would just, summary, reproduction, those adaptive traits are atypical. That's not typical of sea stars, so I think there's something in that to follow up. The larvae clone, is this a symbiosis? It might be independent of food altogether. They might not need food. And this is a bit like what Randy Olson found way back. And the larvae are plastic, and the juveniles themselves are plastic. They've got flexible diet. They can subsist in algae for years, and they have the defensive mechanisms, which we also see in the adults. So I would like to end it there, but I'd like to acknowledge a big team of people that have worked on this, the funding agencies that have given us enough money to get to Lizard Island, and we really do appreciate that. A little bit goes a long way with us. And the COTS teams, if we didn't have the COTS provided by Blue Planet and Inlock, we wouldn't have been doing this work in the first place. So fantastic for the COTS control program. And thank you all for listening.